As humans, everything that happens starting at the moment of conception, through embryonic development and fetal development, and going all the way up to the time when we are born, depends almost exclusively on the ability of the cell to go through very precise cycles of cell growth, DNA replication, and cell division. And this is related in a direct way to what we call the cell cycle. The cell cycle can be viewed as being regulated by a very complex network of pathways that come together to dictate the specific times at which the DNA has to be replicated and the time when the cell has to go into mitosis. But for the purpose of this introductory class in molecular cell biology, the complexity of these pathways will be substantially simplified. In general, the cell cycle can be seen as being divided into two main parts. The first one is mitosis, which is the part of the cell cycle in which the cell actively divides into two new daughter cells. And the second is interphase, which are all other phases or stages of the cell cycle when the cell is not actively dividing. Interphase is further subdivided into three different stages or phases, the most important of them being the S phase. S stands for synthesis, specifically DNA synthesis. So S phase is the time when the cell replicates its DNA. Besides the S phase, there are two other stages which were referred to as gaps, because when they were initially characterized, no specific cellular activity could be associated to those particular phases of the cell cycle. So there is a G1 phase or gap 1 that is located between mitosis and S phase, and then there is a G2 phase that is located between S phase and mitosis. So the cell cycle can be viewed as a continuous progression from one stage to the next of the cell cycle. So the cell cycle can be thought of as starting during cell division. The cell goes then into the G1 stage of the cell cycle, in which time the cell will grow and accumulate nutrients that will allow it to go into the S phase, the time when the DNA will be replicated. And upon completion of DNA replication, the cell will then go into a G2 stage in which the cell will complete the synthesis of all the other components that are required to allow the cell to go into another mitosis. And this cycle is true of all cells in our body except at one very specific time during our lifetime, and that is during embryogenesis. At that time, the fertilized egg will undergo several cycles of cell division without going through real G1 and G2 phases. Notice how, as the cell progresses through the cell cycle, the amount of DNA that it contains will increase. Early in G1, the cell will have its normal chromosomal number of 2n. 2n because we have 2 copies of every chromosome, and that's because we are diploid. At the beginning of the S phase, the 2n number will start increasing because the DNA will start being replicated, and it will continue to increase until when it achieves a final number of 4n at the end of the S phase. And the 4n chromosomal number implies that the DNA is now fully replicated. That 4n number will remain all the way until when the cell ends mitosis. At that time, when the cell splits into 2 new daughter cells, the chromosomal number will go back to be 2n. So the distribution of a cellular population in the cell cycle can be easily determined by knowing the amount of DNA that we have in every cell. The amount of DNA in every cell in a cellular population can be easily established by using a technique known as flow cytometry. In this technique, a fluorescent dye is added to the cells. The fluorescent dye binds to the DNA in a proportional linear manner, so that the more DNA you have in the cell, the more fluorescent dye will bind to the cell. The cells are then run through the flow cytometer, which has the ability to detect the volume of the cell based on the amount of light that is scattered by every cell, and has also the ability to quantify the amount of fluorescence that is emitted by every cell. The final measure that is given by the flow cytometer indicates the proportion or percentage of cells in the cellular population that exhibit different amounts of fluorescence. The intensity of the fluorescence is in the x-axis, whereas the percentage of cells emitting that particular intensity of fluorescence is in the y-axis. Remember, the DNA concentration will be determined by the intensity of the fluorescence. The profile presented in this particular figure represents the typical profile of any mammalian cell line maintaining tissue culture. The biggest peak corresponds to the cells that are in the G1 stage of the cell cycle. The second larger population is represented by the cells that are in the S stage of the cell cycle. Finally, the smallest fraction of cells correspond to those that are in the G2M stage of the cell cycle. These two stages of the cell cycle overlap in the figure simply because they correspond to cells that contain the same amount of DNA, that is 4N. So it is impossible, based on this kind of analysis, to separate cells that are in the G2 from cells that are in the M phase of the cell cycle. The main factor that dictates the percentage of cells that are present in every one of the different phases of the cell cycle is the duration of that particular stage of the cell cycle. So the longest stage in the typical mammalian cell line is the G1 stage, followed by the S phase, and followed by the G2 and M phases. Passage through the cell cycle is a process that is very tightly regulated, and that tight regulation allows the cells to accurately transmit the genetic information to the new daughter cells that will be produced. In addition to that, the tight regulation of the cell cycle allows the cell to maintain the normal chromosomal number, also known as ploidy. And in mammalian cells, that number is a diploid number. So the maintenance of diploidy requires a tight regulation of the passage through the cell cycle. Errors in the regulation of passage through the cell cycle can result in alterations in chromosomal number, known as aneuploidy. So cells that exhibit aneuploidy are cells that have altered chromosomal numbers. This is a condition very frequently found in cancer cells. There are 5 main types of regulatory signals that control passage through the cell cycle. The first one is the size of the cell. The signal plays a role in the control of passage through the cell cycle in mammalian cells, but a very minor one. However, it is the most important signal that controls passage through the cell cycle in lower organisms such as fish and yeast. The presence or absence of nutrients is another important regulatory signal that determines passage through the cell cycle. And again, in another type of lower organisms, the budding yeast, also known as Saccharomyces cerevisiae. This is the main signal driving passage through the cell cycle. In these lower organisms, the step that determines whether the cell will be committed to continue in the cell cycle is the so-called start point. That is, passage through this particular point in the cell cycle commits the cell to continue going through the cell cycle. Importantly, this start point is located in the G1 stage of the cell cycle. In higher eukaryotic cells, such as in mammalian cells, the main signal that dictates passage through the cell cycle is the presence or absence of growth factors. So cells require the continuous stimulation with growth factors in order to progress through G1 and go into S phase. The point that dictates commitment to passage through the cell cycle in mammalian cells is known as the restriction point. Cells that don't go through the restriction point can exit the cell cycle and go into what is known as a quiescent stage, known as the G0 stage of the cell cycle. And even though the cells that are in the G0 stage are considered to be quiescent from the replicative point of view, from the metabolic point of view, the cells that are in G0 can be very metabolically active. So the fact that a cell line is in the G0 stage doesn't mean that it is not metabolically active. A different type of signals that also regulates passage through the cell cycle is known as the checkpoints. Checkpoints are times during the cell cycle in which the cell checks for the presence of a specific type of signals, and if activated, the cell is stuck in that particular stage of the cell cycle without being allowed to continue forward. In mammalian cells, there are 4 different checkpoints, and any one of them has the ability to stop transition through a cell cycle. 3 of the checkpoints check for DNA damage, whereas 1 of the checkpoints checks for the proper alignment of chromosomes at the mitotic spindle. Finally, the last type of signal that regulates progression through a cell cycle is one that prevents the cells from replicating the DNA more than once per cell cycle. This mechanism involves the activation of the helicase that is responsible for opening the double-stranded DNA during DNA synthesis, during DNA replication, and that is MCM. So NCM, the helicase responsible for opening the double-stranded molecule during DNA replication during S phase, is active only once per cell cycle, so that the DNA will be allowed to be replicated once and only once per cell cycle. And with this, we finish our introductory movie on the regulation of cell cycle. Watch it as many times as you feel you need it. Thanks.